Bismillahirrahmanirrahim. Assalamu alaikum dear students. Welcome to your channel The Anatomy Canvas. In today's class we are going to start with the histology of the nervous system. So learning objective for the today's class are that the function of the nervous system, the organization of the nervous system, nerve tissue and nerve cell types, structure of the neuron and structure of the chemical synapses. So what is nervous system? It is defined as like CPU of the computer. The nervous system is the master controlling system of the body. It is designed to constantly and rapidly adjust and respond to the stimuli the body receives. It includes brain, cranial nerves, spinal cord and associated peripheral nerves. So division of the nervous system are either central nervous system or peripheral nervous system, as you all know. The central nervous system consists of brain and spinal cord, and peripheral nervous system consists of the cranial nerves and the spinal nerves along with the ganglia and the receptors as well. So properties of the neurons are: first, they have a property of excitability, that is irritability, the ability to respond to the environmental changes or the stimuli. Second property, they have the property of conductivity. They respond to the stimuli by initiating the electrical signals that travel quickly to other cells at distant locations. Then the secretions: upon arrival of the impulse at a distant location, the neurons usually secrete a chemical neurotransmitter at a synapses that cross the synaptic gap and stimulate the next cells. So these are the different properties of the neuron: the excitability, the conductivity and the secretion. So neurons, what is a neuron? Neuron cell properly have three parts: the cell body or the soma, then the dendrites and then the axon. So different branches: the cell body or the soma, also called the perikaryon. And this is the histological slide of the neuron. Here you can see the neuronal cell body, its processes called dendrites and also the axon. So we will discuss all of these things in detail now. Nerve tissue consists of the two major types of the cells. The one are the neuron and other are the glial cells. Neurons are responsible for conduction, propagation and reception of the nerve impulses. The processes called axons or dendrites extend from these cells. The glial cells, also called the neuroglial cells, are associated with the neurons. They have no axons or dendrite, and these cells are involved in the nutrition, the support, the insulation, the protection of the neuron. So these are the cells which are basically involved to maintain the physiological conditions of the neurons. They help the neuron in their nutrition, support, insulation and etc. other things which are important for the neurons. And basically neurons are the cells which are responsible for conduction, for propagation of the nerve impulses and reception of the nerve impulses. So CNS composed of the neurons are the true conducting cells in nervous tissue and neuroglial cells, two types of cells. In central nervous system, the glial cells are the astrocytes, the oligodendrocytes, microglia and ependymal cells, and in peripheral nervous system they are the Schwann cells and the satellite cells. So this is the diagram of the neuron. Here you can see this is the cell body of the neuron, also called the soma, also called the perikaryon. This is the conical part called axon hillock, and this part is the segment which is called initial segment. This is the actually part where the action potential is generated, and then the axon starts, and in this axon is myelinated, in which you can see multiple node of Ranvier. Then the axon ending where synaptic buttons and synaptic vesicles are present, and this is the presynaptic membrane, the synaptic cleft and postsynaptic membrane. And here you can see these are the dendrites. So first we will study the perikaryon. The perikaryon, the nerve cell body, contains a nucleus and typical cell organelles. The nucleus is the large, it is central in most of the cells. A large amount of chromatin is present, which show intense synthetic activity, means this cell is highly anabolic cells. It has prominent nucleolus which gives its owl's eye appearance. It has body in females, which is the dominant X chromosome in the females. Also, the rough endoplasmic reticulum is present. Large amount of rough endoplasmic reticulum is present for the synthesis of structural and the transport protein. The nasal body are the substance seen with the light microscopy are condensation of this rough endoplasmic reticulum and the free ribosomes. So here is the histological section of the neuron. You can see this is the neuronal cell body, in which you can see the nucleus along with the nucleolus is present, and in the cell body or the perikaryon, this darker color area show large amount of the rough endoplasmic reticulum, either in the form of the nasal granules or present as the stacks. These are the processes of the neuronal cell body. These may be dendroids, may be axons. In H&E stain section, it is difficult to differentiate between the axons and the dendrites. This is another diagram showing you the histological section of the neurons, and in this cell, this is the perikaryon of the neuronal cell. Here you can see a prominent nucleus with the prominent nucleolus, which gives a typical owl's eye appearance, and these all are the nasal granules, which are aggregates of the rough endoplasmic reticulum. This is another section of the neuron in which you can also see the nucleus with the nucleolus and the nasal granules are present, and these are the smaller nuclei, which may be the nuclei of the glial cell, which are present in the substance of the nervous tissue. So next important component of the perikaryon is the Golgi complex, only found near the nucleus in perikaryon, that is perinuclear in position, expected since intense synthetic activity of the neurotransmitters and neurohormones that must be packaged in the vesicles. Also important from high anabolic activity is the number of the mitochondria, so the cells have abundant for the high energy requirements. They also have large amount of the neurofilaments. These are nearly intermediate type of the filaments, which are 10 nanometer in size. Also contain large amount of the microtubules, which are important in transport of the materials, that is the neurotransmitters, in the either in antegrade or in retrograde directions. Also have inclusion bodies. The pigment deposit functions not known. Most important are the lipofuscin deposit. These are residual bodies from the autophagosomal activity, and they increases with it. This is again your MCQ as well. So cell processes, next is the dendrite. The cell process extending away from the perikaryon that may be branched. They form the receptive area for the synaptic contact from the other neuron. So this is again your MCQ, that the receptive area for the neuron is the dendrite. Dendrites form from the larger neurons, may be lightly myelinated, but usually they are unmyelinated. Cytoplasm in these processes similar to that of the perikaryon, that is free ribosomes, nasal granules, occasionally Golgi bodies present. So this is a very important point to differentiate on electron microscopy between the dendrite and the axons. The dendrite may contain the free ribosome, the nasal granules, and occasionally the Golgi bodies. But in case of axons, no free ribosomes, nasal granules, or Golgi bodies will be present in axon. So this point help us to differentiate between the dendrite and axon in the electron microscopic pictures. So this is the diagram of the neuron. Here you can see the neuronal cell body, also called the perikaryon. This is the nucleus and the nucleolus. The cytoplasm contain large amount of the nasal granules. Also you can see the large number of the mitochondria are present. These are the small dendritic process, and here you can also notice that in these dendrites the nasal granules are also present. This part is the axon hillock, then the initial segment where action potential will be generated, and then the axon starts. This axon is myelinated. Here you can see the node of the Ranvier. Also the axon's collateral branch is given over here. This is the section of the axon in which you can see these are the neurofibrils which are present, which are surrounded by the myelin sheath, which is produced by the Schwann cell, and this is the neurylem of the Schwann cell. So axons are one long thin process extending away from the perikaryon. It may be branched. Ends of the branches form synapses with other neurons or the muscle cells. It may be unmyelinated or may be myelinated. In case of central nervous system, myelination takes place by oligodendrocytes, and in case of the peripheral nervous system, the myelination is done by the Schwann cells. The axon structure is that it has an axon hillock, which is a pyramidal or conical shape region associated with the perikaryon where axons begins. Cytoplasm in this region lacks the ribosomes and the organelles. However, neurofilaments and microtubules are present. Then the initial segment may be myelinated. Initial segment is usually unmyelinated portion of the axon between axon hillock and beginning of myelination. Remainder of the axon may be myelinated. It may be branched. The axons carry electrical impulses to synapses at the end of the axon, and except for the synapses, the axon cytoplasm has few organelles, not much synthetic activity in that part of the neuron. The microtubules and microfilaments are present in axon, and axonal transport can be antegrade, that is normally the organelles transported from the cell body towards the periphery. It can be retrograde in case of the toxins or viruses which are carried from the periphery to the neuronal cell body for the process of the detoxification. So axon, another point is that they have side branches called collaterals. They end in a fine process called the axon terminal. So this is the collateral branch, and these are the fine processes which are called the axon terminals. They have the swollen tips which are called synaptic end bulbs. Here you can see synaptic end bulbs over here, and these synaptic end bulbs contain the vesicles which have the neurotransmitter. So the synapses along the length of axon results in varicosities or swellings which are called the synaptic end bulbs, and they are called the button end passage. The synapses at the end of the axon or axon branches are also swollen into club shape called the button terminux. So here you can see the button terminux at the end. Now we study how we classify the neurons. So first we classify on the number of the processes found on the cell body, that they may be multipolar, they may be bipolar or unipolar. Multipolar have several dendrites and one axon. The most common cell types are the multipolar. Most of the motor neurons and the interneurons are multipolar in nature. Bipolar neurons have one main dendrite and one axon. The true examples of bipolar neuron in human body are found in the retina and inner ear, where they form the bipolar neurons. The unipolar neurons, the one processes which then divide into two, one peripheral and one sent to the center of the central nervous system, and unipolar neurons also called unipolar, pseudo-unipolar, because they were actually bipolar developmentally. All the sensory neurons are the pseudo-unipolar. Bipolar neuron, this is an example of the bipolar neuron. One main branch, this is dendrite, and this is the axon. They are found in the retina and the inner ear. The unipolar neurons have one process that divide in a T-shaped manner, and one is peripheral process and other goes to the center. So various type of the neurons have classification on the shape and morphology, that they may be multipolar, means more than two processes. They may be bipolar, or they may be unipolar or pseudo-unipolar. Neurons may be multipolar. So in this diagram you can see this is the pseudo-unipolar neuron, this is the bipolar neuron, and these are different multipolar neurons. Functional classification of the neurons is that they may be sensory neurons, they may be interneurons or motor neurons. So sensory neurons or efferent neurons are specialized to detect stimuli and transmit the information to the central nervous system. They begin in any organ in the body but end in brain and the spinal cord. The interneurons are also called the association neuron. They lie entirely in the central nervous system. They receive signals from many different neurons and perform an integrative function, that is decision making to respond to the different stimuli. And motor neurons are efferent neurons, transmit the appropriate response from interneuron to an end organ, that is muscle or the gland cell. So this is the functional classification. They may be sensory or efferent, they may be interneurons or association neurons, and they may be motor or efferent neurons. Then the functional classification of neurons based on direction of the conduction, again sensory or motor or interneuron. So sensory conduct towards the central nervous system more than 100 million neurons. The motor neurons conduct away from the central nervous system more than 5 lakh, and interneurons interpose between sensory and motor. There are large number of the neurons, more than 500 billion neurons are interneuron in nature. So they may be named according to the histologist who first described them, or according to their appearance, like Purkinje cell, which was first described by the scientist named Purkinje, or they may be pyramidal shape according to the shape of the perikaryon, which is pyramidal in shape. So classification can also be based on length of the axon, that is Golgi type 1 neuron and Golgi type 2 neuron. Golgi type 1 have a very long axon, for example the pyramidal cells of cerebral cortex and anterior horn cells of the spinal cord. The Golgi type 2 neurons have a short neuron. Mostly the interneurons are Golgi type 2s. Most of the time they are also inhibitory in nature. So this is the slide, this is the model which is present in your histology in your museum for your OSPE. So look at this model. This is the section of the neurons. Here you can see the nucleus along with the nucleoli which are present. This is the nuclear membrane. Here you can see the Golgi apparatus and the rough endoplasmic reticulum which is present over here. Large number of the mitochondria are present. The neurofilaments are present. This is the axon hillock, and then the myelinated axon terminal is present. These are the different dendrites are present in the axon hillock. These brownish color areas shows the nasal granules. In this section you can see this is the axon which is covered by myelin sheath. So this is the myelin sheath which surrounds and it is formed by the Schwann cell. So this is the basically model which is present in your museum. This is another model which is present in your museum for the OSPE. This is the section of the neuron in which you can see this is the nucleus, and it contains nuclear membrane, contains many nuclear pores. Also the nucleoli are present. In the cytoplasm you can see large number of the nasal granules. These are the axon dendrites, and this is the axon where you can see neurofilaments as well. And this is the section of the nerve fiber, the axon in which axon terminal is present over here, and surrounding these are the myelin sheath which is formed by the Schwann cell. This is the nucleus of the Schwann cell, and this is the node of the Ranvier. This is another model in which you can see the synapses of the motor neuron with the skeletal muscle. So this area is the presynaptic membrane, and here in this area you can see large number of the synaptic vesicles are present which contain the neurotransmitter, usually in this case the acetylcholine. Then this black color space is the small synaptic cleft, and then this white color is the postsynaptic membrane which is formed by the sarcolemma of the skeletal muscle. So this is the section of the skeletal muscle, and this is the neuron. This is another model which is present in our lab and museum. So here you can again see the synapses. This is the site of the skeletal muscle, and here you can see nerve fiber. So this area is the presynaptic membrane, this white color, in which you can see in the in the cytoplasm you can see large amount of the presynaptic vesicle as well as the mitochondria are present. The blue color area shows the synaptic cleft and then the postsynaptic membrane, and this is the axon which is in yellow color. This is the axon, this is the myelin sheath which is formed around it. This is the nucleus of Schwann cell. Here you can also see the nuclei of the Schwann cell. So this is the enlarged view of the same model, in which you can see the presynaptic membrane, the white color synaptic vesicles, the blue color synaptic cleft, and this area is postsynaptic membrane formed by the sarcolemma of the skeletal muscle. So now we move on to the nerve structure. The nerves are only in the periphery. They are cable-like arc in the peripheral nerve system. They may be cranial nerve, they may be spinal nerve. They consist of hundreds to lakhs of myelinated and unmyelinated axons. The endoneurium surround each axon, that is the nerve fiber. These nerve fiber, when combined, they form the nerve vesicle, which is formed by the perineurium, and when these vesicle combine, they form the nerve bundle, and this nerve bundle is surrounded by the epineurum. So conduction in these neurons is the saltatory, that is jumps from one node of Ranvier to other node of Ranvier in myelinated nerve, and continuous in cases of the non-myelinated neurons. So this is the section of the neuron. Here you can see the axons which are present, and these axons are covered by very fine connective tissue, that is the endoneurium. These axons collectively form the nerve vesicle, and this nerve vesicle is surrounded by the perineurium, and these vesicles, when they combine, they form the nerve fiber or entire nerve, which is covered by the epineurum. So this is electron microscopic picture of the nerve fiber, in which you can see different axons are present, and these axons are covered by endoneurium, and here you can see the vesicle which is covered by the perineurium. These are the blood vessels which are present in perineurium, and all these vesicles combine to form the nerve fiber which is covered by the endoneurium. Again, the H&E stain section of the nerve fiber, in which you can see these are the axons which are present, and these axons, this is the cross section of the nerve fiber. These axons are covered by endoneurium, and this is the nerve vesicle which is covered by perineurium, and these all are the nerve vesicles. So in this diagram you can't see the epineurum. This is another diagram at lower magnification, in which you can see multiple vesicles which are present, and these vesicles combine to form the nerve fiber which is covered by the epineurum. These vesicles are covered by the perineurium, in which individual nerve fibers are present. This is the perineurium, the connective tissue which contains blood vessels. This is the slide which is present in your histology lab, and this is the cross section of the nerve fiber, and this one is the longitudinal area, longitudinal section of the nerve fiber. So this is slide from your histology lab. In this you can see smaller axonal fibers are present in these areas which are covered by endoneurium, and this whole vesicle is covered by the perineurium. So these are the longitudinal muscle fiber, the longitudinal section of the nerve fiber. This is another slide, basically a slide of the ganglia, in which you can see neuronal cell bodies are present, and in these areas you can see the cross section of the nerve fibers which are present in it. This is again slide from your histology lab. So here are the references: the basic histology by Laikus and Siddiq, the histology text and atlas by Janquera, and histology text and atlas by H. And here I complete my today's class. Thank you very much, dear students, for your attention for the today's class. Allah Hafiz till the next class.